Modern biology is now a big data science. A single sequencing run, a proteomics experiment, or a metabolomics study can generate an enormous amount of information. And this data is coming from every layer of biology. We may collect genomic sequences, RNA expression profiles, protein structures, mutation data, metabolite measurements, or clinical records. So the challenge is no longer only generating data. The challenge is managing it in a meaningful way. Raw data by itself is not very useful. If it is not organized, searchable, and linked to biological context, it becomes difficult to interpret. If you look at the idea on the slide, the key message is that biology is producing information at a scale that cannot be handled manually. That is why we needed a digital system to preserve, organize, and explore this biological knowledge. So how do we build that system? A biological database is much more than a storage device. It is an organized and searchable repository designed specifically for biological data. That means it does not just hold information. It also helps researchers retrieve, filter, compare, and analyze it efficiently. For example, instead of manually searching through thousands of papers for one protein, a database allows you to find the sequence, function, structure, and related literature in seconds. Biological databases can store many different kinds of information. Some store DNA or RNA sequences. Some store protein functions. Others store 3D structures, metabolic pathways, disease associations, or drug interactions. If you look at the concept on the slide, the key idea is that these databases act like a digital ark. They preserve biological knowledge and make it accessible. But to use them effectively, we need a way to organize them conceptually. That brings us to the architecture of biological data. Biological databases are often organized according to the flow of biological information. This follows the central dogma: DNA to RNA to protein, and then onward to phenotype, pathways, and disease. That structure makes sense because each layer answers a different type of biological question. DNA databases tell us about the genetic blueprint. RNA databases help us understand gene expression. Protein databases tell us about molecular function. Pathway and interaction databases help us see how molecules work together. And clinical databases help connect all of this to observable traits and disease states. If you look at the slide, think of this as a layered map of biology. Each type of database captures one part of the system. So let us begin at the foundation, the genetic code itself. At the most fundamental level, biology begins with sequence information. That is why nucleotide databases are among the most important biological resources. Databases such as GenBank, ENA, and DDBJ store DNA and RNA sequences submitted by researchers around the world. These are primary repositories for sequence data. But storing sequence alone is not enough. Researchers also need to visualize where genes are located and how the genome is organized. That is where genome browsers such as Ensembl and UCSC Genome Browser become important. They act like interactive maps of chromosomes. They show genes, exons, introns, variants, and regulatory regions in genomic context. If you look at the slide, the important idea is that these resources allow us to move from raw sequence to genomic organization. But genes are only the instructions. The actual molecular work of the cell is often carried out by proteins. If DNA is the code, proteins are the molecular machines built from that code. They perform enzymatic reactions, transport molecules, regulate signaling, and carry out structural roles inside the cell. That is why protein databases are so important. UniProt is one of the major databases for protein sequence and functional annotation. It tells us what a protein is, what it may do, where it acts, and what kind of domains or motifs it contains. Then we have the Protein Data Bank, or PDB, which stores the three-dimensional structures of proteins and other macromolecules. This is critical because structure often determines function. If we know the shape of a protein, we can better understand how it binds ligands, performs catalysis, or interacts with drugs. If you look at the concept on the slide, protein databases help us move from sequence to function and from function to structure. But just because a gene or protein exists does not mean it is active in every cell. To understand activity, we need expression data. The genome gives us the full set of possibilities, but gene expression tells us which genes are actually being used in a particular cell or condition. This is very important because cells with the same genome can behave very differently depending on which genes are active. Databases such as GEO, the Gene Expression Omnibus, store transcriptomic data from microarrays and RNA-seq experiments. These databases allow researchers to compare expression patterns across tissues, diseases, treatments, and developmental stages. For example, we can compare which genes are upregulated in tumor tissue relative to normal tissue. That kind of comparison helps identify disease-associated pathways and biomarkers. If you look at the slide idea, the key message is that expression databases provide a real-time molecular snapshot of cellular activity. But beyond gene expression, there is another layer of control that influences how genes are read. Not all gene regulation comes from changes in DNA sequence. Cells also use epigenetic modifications to control how genes are turned on or off. These include DNA methylation, histone modifications, and changes in chromatin accessibility. This layer of biology is studied through epigenomics databases and projects such as ENCODE. ENCODE has been especially important in cataloging regulatory elements such as promoters, enhancers, and transcription factor binding regions. This helps researchers understand not just what genes exist, but how their expression is controlled. If you look at the slide, the important idea is that epigenomics acts like a regulatory layer on top of the genome. Now, even with genes and regulatory elements identified, biology still does not happen in isolation. Molecules work together in networks. A cell is not just a collection of separate molecules. It is a highly connected system. Genes, proteins, metabolites, and signaling molecules constantly interact with one another. That is why pathway and interaction databases are essential. Resources such as KEGG and Reactome help map biological pathways. They show how molecules participate in metabolism, signaling, and disease-related processes. Then, databases such as String help identify protein-protein interactions. This is important because sometimes the function of a protein becomes clear only when we see what other molecules it interacts with. If you look at the slide, think of these resources as the network map of the cell. They help us move from isolated genes to coordinated biological systems. But among all individuals, one very important source of biological variation is the genome itself. Even though humans share most of their genome, small differences in sequence can have major biological consequences. One of the most studied forms of variation is the single nucleotide polymorphism, or SNP. This is a change in just one nucleotide position in the genome. Databases such as dbSNP catalog these variations. These resources are important because some variants are harmless, while others can influence protein function, disease susceptibility, or drug response. In some cases, even a single nucleotide change can disrupt a protein or alter a regulatory region. If you look at the slide, the key idea is that small sequence changes can create meaningful biological differences. When those differences begin to affect traits or disease, we move into phenotype databases. A phenotype is the observable outcome of genetic and biological processes. That may include a disease, a trait, a clinical condition, or a developmental abnormality. Databases such as OMIM or online Mendelian inheritance in man help connect genes with human diseases. This is extremely valuable because it links molecular biology to clinical reality. Instead of only asking what a gene does, we can ask whether mutations in that gene are associated with a known disorder. If you look at the slide, the key message is that phenotype databases help bridge the gap between laboratory biology and human health. One area where this connection becomes especially complex is cancer. Cancer is fundamentally a disease of genomic and molecular dysregulation. Tumors often contain many mutations, altered signaling pathways, copy number changes, and expression abnormalities. That complexity requires specialized databases. One of the most important examples is TCGA, the Cancer Genome Atlas. Resources like TCGA collect large-scale data across many tumor types, including genomic, transcriptomic, epigenomic, and clinical information. This allows researchers to study cancer not just as one disease, but as many biologically distinct subtypes. If you look at the slide, the key message is that cancer genomics databases help decode the molecular chaos of tumors. Once we identify disease-related targets, the next question becomes, how do we intervene therapeutically? Biological understanding becomes especially powerful when it can guide treatment. Drug and chemical databases help connect biological targets with therapeutic molecules. A resource like DrugBank maps drugs to their known protein targets, mechanisms of action, and pharmacological properties. This is extremely important in drug discovery and translational research. If we know which protein is involved in disease, and we know which compounds interact with that protein, we can begin to design or select therapeutic strategies more effectively. If you look at the concept on the slide, These databases help bridge molecular biology and pharmacology. But to understand whether a biological system is truly functioning normally, we often need to look at its final biochemical output. If the genome tells us what could happen, the metabolome tells us what is actually happening at the biochemical level. Metabolomics focuses on small molecules such as amino acids, lipids, sugars, and metabolic intermediates. These molecules reflect the real-time state of cellular activity. The Human Metabolome Database, or HMDB, is one of the major resources in this field. It provides information on human metabolites, their structures, biological roles, and disease associations. This makes metabolomics especially valuable for studying physiology, disease states, and treatment responses. If you look at the slide, the key message is that metabolomics captures the biochemical outcome of cellular function. But all of this data still needs interpretation, context, and scientific explanation. That brings us to literature databases. Data becomes much more useful when it is connected to published scientific knowledge. That is the role of literature databases such as PubMed. PubMed indexes a massive body of biomedical research literature and allows scientists to connect genes, proteins, pathways, and diseases with peer-reviewed studies. This is critical because databases may tell us what is associated with a molecule, but the literature helps explain why and how. If you look at the slide, literature databases act like an intellectual bridge between raw data and scientific interpretation. They provide the evidence base that supports functional annotation, pathway interpretation, biomarker validation, and clinical relevance. As biology moves forward, the most powerful discoveries will not come from one database alone. They will come from integration. No single biological database can answer every question. Each one captures only one layer of biology. The future of biological research lies in integration. We need to connect genomics with transcriptomics, proteomics with pathways, variation with disease, and drugs with molecular targets. That is the foundation of systems biology. It allows us to move beyond isolated observations and begin understanding the organism as a connected biological system. If you look at the final concept on the slide, the important message is that the power of modern biology comes not only from data generation, but from linking data across multiple biological levels. That is why biological databases are so important. They are not just storage tools. They are the digital foundation of modern life science research. Thank you.